Bismillahirrahmanirrahim. This is Dr. Nighat Aslam from Department of Biochemistry. This is my second lecture on molecular biology techniques. In my previous lecture, we studied about the restriction about enzymes and the DNA. What is the DNA cloning? Now, in this lecture, we are going to start with the vectors, with the type of vectors. These vectors, they are used in the DNA cloning techniques. What is vector? This is a molecule of DNA to which fragment of DNA to be cloned is joined. There must be three properties present in the vectors in order to be made the vectors useful. They must be capable of autonomous replication. They must have restriction site so that we can cut the vectors and join our DNA of interest in these vectors. And it must have the antibiotic resistance gene. This resistance gene, antibiotic resistance gene, will help the vector to be selected, that which vector contain our gene of interest and which vector do not contain our gene of interest. This antibiotic resistance gene help to select the recombinant vectors. The first type of the vector which we are going to study is a plasmid. Plasmids are small circular duplex DNA molecules whose natural function is to confer antibiotic resistance gene to the host cell. They are naturally present in the bacteria, and in the lab we isolate these plasmids from the bacteria. We can also prepare these plasmids in our lab. For example, this PBr 322 is a plasmid which is prepared artificially with our required properties in lab. Plasmids, prokaryotic organisms, they also contain a large circular chromosome, but these plasmids, they are in addition to that DNA. Up to 15 kb of the fragment, they can be ligated, like 15 kb of the DNA, our gene of interest, they can be ligated. The plasmids which can be propagated in the cells of two or more different species, they are called the shuttle vectors. Means the vectors, first we change these vectors into the recombinant vectors. They are grown or they are allowed to transform in the bacteria. They are replicated there. Then we isolated those vectors from bacteria and then transform into some other vector, some other host. And these vectors which can propagate in two or more different hosts, they are called shuttle vectors. PBr 322, it is I have told you that it is artificially made plasmid, and they are routinely constructed, which contains origin of replication, some antibiotic resistance genes, and some routine restriction sites. These are the two antibiotic resistant genes, ampicillin resistant gene and tetracycline resistant genes, and these are the XOR1, XOR2, PST1, BAMH1, BAM3, HINDI3. They are all the restriction sites which are used to cut open the plasmid so that we can insert our gene of interest in the plasmid. The second type of vectors are the phages. They are the bacterial viruses, bacteriophages. They are also called the bacteriophages. They are the viruses which attack on the bacteria, so they are called bacteriophages. Their DNA, they can act as vectors. They usually have linear DNA molecules into which foreign DNA can be inserted at several restriction enzyme sites. They can accept the DNA fragment with more longer than the plasmids, about 10 to 20 kb long. They can be inserted. The DNA fragment about 20 kb long can be inserted into the bacteriophage viruses. A limitation is imposed by the amount of the DNA that can be packaged into the phage head. Another type of vector is the cosmid. Cosmid combines the best feature of plasmid and phages. Mids is from the plasmid, and cosmids, the cos is from the cos sites required for the packaging lambda DNA into the phage particles. So it combines the best features of the plasmids and phages. These vectors grow in the plasmid form in bacteria, but since much of the unnecessary lambda DNA has been removed, about 35 to 50 kb long, it has been removed, and more chimeric DNA can be packaged into the particle head. So longer DNA can be ligated. Another types of vectors, they are the bacterial artificial chromosomes, BAC, yeast artificial chromosomes, YAC, and E. coli bacterial P1 based PAC vectors. These vectors, they accept and propagate DNA inserts of several hundred kilobase or more, and have largely replaced the plasmid, phage, and cosmid vectors for some cloning and gene mapping applications. So these are the different types of the vectors, and they are the DNA size which they can accept. Any of the vector, they can be used in our experiment in the lab according to our needs and according to the requirement of the experiment, that how much amount of the vector, how much amount of the DNA can be inserted, or whether these vector, they can be propagated in different hosts, they can be propagated in the expression host, or any of the limitation which can be help in choosing the what is the best vector to be used in our experiments. This is the vector called PBR322, plasmid called PBR322. Here you are seeing that there is the ampicillin resistance gene. This is the ampicillin resistance gene, and where we are going to insert our DNA of interest. This site contain antibiotic resistance, contain the restriction enzyme site PST1. We will cut open this vector by using this enzyme PST1, and by using same enzyme PST1, same restriction enzyme, we will have our DNA, we will cut our DNA which we want to insert. So their ends, they can be, we use the same enzyme to cut open the vector, we will use the same enzyme so that we have the sticky ends, the same sticky end which can be ligated into this host. So now what happened when we cut this plasmid by the enzyme PST1 and the DNA is ligated, this site, ampicillin resistant site, this will be disrupted, and this ampicillin resistant enzyme plasmid will be changed into the ampicillin sensitive plasmid. So this chimeric PBR322 plasmid, if it is grown in the media which contain ampicillin, if it is died or if it is disrupted, if its growth cannot be propagated, then it means that this plasmid is sensitive to the ampicillin, and this plasmid contain our DNA of interest. We will go backward and pick the colonies or pick the bacteria from which we took this sample, and will choose this and choose the samples to be further used in experiments. So our chimeric PBR322 will be sensitive to the ampicillin drug, which shows that this contain our DNA of interest. These are the different types of enzymes which are used in the recombinant DNA research. Phosphatases, they will dephosphorylate, and it is used in the to prevent the self-ligation. DNA ligase, DNA polymerase, DNase 1, exonucleases, these are all enzymes which we have studied several times in our lectures. Just give a single look, and you must have the idea that what these enzymes do and what is their application. Just give a single look at these tables from the Harper. DNA library. This is a picture of Petri plates which contain this media, a yellowish media, on which these small colonies, they are grown. They are bacterial colonies. This is I took this picture I took from the internet, so as to show that what type of growth the bacterial colonies have on the Petri plates. What are the DNA libraries? DNA library is a collection of cloned restriction fragments. This is a type of, this is a DNA DNA library. It shows a collection of cloned restriction fragments into bacteria, which was spread over the media, and this shows the growth. So the combination of restriction enzymes and various cloning vectors allows the entire genome of an organism to be individually packed into a vectors. What we do, that we took the entire genome, we isolate the DNA, we use the restriction enzyme to cut that DNA into fragments, and then these fragments, they are ligated into the plasmid, and these plasmids, they are transformed into the bacterial cells. Then these bacterial cells, they are grown, and we have the genomic DNA library. This is a collection of cloned growth of the bacteria. So genomic DNA library is prepared from total DNA of cell lines of the tissues. We just take the total DNA and use the restriction enzyme, ligate into the vectors, and transform into the bacteria and get the growth. Often prepared by performing partial digestion of total DNA with restriction enzyme that cuts the DNA frequently, example for base cutter such as the TAC1. So we use the partial digestion, that we do not give the full time to totally digest the DNA. We just give half of the time, so that it contain frequent cuts on the DNA. BAC, YAC and P1 vectors, they are preferred since they can accept very large fragments of DNA. So this is the genomic DNA library. Another type of library is the cDNA library. cDNA library, if a protein coding gene of interest is expressed at a high level in a particular tissue, the mRNA transcribed from that gene is likely also present at high concentration in the cells of that tissue. What we do, we take the mRNA. We start our experiment by isolating the mRNA from that tissues which contain the proteins of that mRNA. For example, if we take reticulocyte, the mRNA is composed largely of the molecules that code for alpha globulin and beta globulin chain of hemoglobin A. So this mRNA can be used as a template to make a cDNA molecule. So starting with see the figure which is at the left hand side, the upper, we how we change this mRNA into the cDNA. First we will start our experiment starting from the mRNA. mRNA, you are seeing that it contains the poly A tail at the 3' end. We use this mRNA as a template, and first of all we use the reverse transcriptase enzyme, and all the nucleotides ATP, DCTP, DGTP and DTTP. By using these nucleotides and reverse transcriptase, mRNA as a complementary strand, as a template strand, and the complementary strand is made. This is called the complementary DNA. Now by using the alkali, the RNA is cleaved. This blue strand, it is cleaved. Then again by using the DNA polymerase, not the reverse transcriptase. In this time we are using DNA polymerase, and again DNTPs, this fragment, the complementary DNA fragment, it is synthesized. Now by using the DNA ligases, we join these fragments and a full double stranded DNA it is formed. This double stranded DNA, it was obtained from the RNA. This is called the complementary DNA. And this complementary DNA, it will be ligated into the vector, and this vector, it will be transformed into the bacteria, and the bacteria, the growth which we will obtain will called as cDNA library. So the cDNA library comprises complementary DNA copies of the population of mRNA in a tissue. Human insulin is prepared. Therapeutic human insulin is made in the bacteria through this technology. In some proteins, co and post translational modifications, they are also required. For that we use eukaryotic and even the mammalian host, but some of the time these co and post translation modifications are not required. Bacterial host, they can also be used. Now sequencing, how the clone DNA fragments, they can be sequenced, or any of the DNA fragment, how they are sequenced, how human genome project was completed. This shows the basic principles and underlying procedures which was used by the human genome project. How we can sequence a part of the DNA. First of all, we took the single stranded DNA of unknown sequence. We don't know what is its sequence. In the first step, single stranded DNA of unknown sequence is used as a template. Then we use a primer. This is a primer. Add primer, DNA polymerase, the enzyme which will synthesize its complementary strand, and the DNTPs, DATP, DGTP, DCTP and DTTP. Now in the next step, split the sample into 4 tubes, each with one of the 4 dideoxynucleotide. We split these samples into 4 tubes, and in every tube, in the first tube we will add the dideoxy GTP, in the second tube we will add dideoxy ATP, in the third tube we will add dideoxy CTP, in the fourth dideoxy TTP is added. Synthesis proceeds until the dideoxynucleotide is incorporated into the DNA strand. Because the dideoxy contain no 3' hydroxyl group, incorporation of this dideoxy nucleotide terminates the elongation at that point. DNA terminating in the dideoxynucleotide cannot be elongated because it lacks the 3' hydroxyl, which is required for the formation of 3' to the 5' phosphodiester bond. So this is the division of our experiment into the 4 tubes, each containing the separate dideoxynucleotide. So the first one shows that this is the dideoxy GTP, and it will synthesize its strand, but whenever the DNA polymerase use the dideoxy G, its chain cannot be further elongated. So this tube, it will contain 2 types of the strand. In the first, it will be terminated earlier, in the second it will be elongated further if the normal GTP was inserted before. In the second, this contain as this contain the dideoxy ATP, it will synthesize unless the DNA polymerase use the dideoxy A. And in the third, this chain will be synthesized up till here, and again the dideoxy TTP tube will also contain two types of the fragments. In the next, what we will do, that we will do the polyacrylamide gel electrophoresis. There are 4 types of the samples. They are allowed to run on the electrophoresis, and in the first gel we will have the 2 types of the fragments. The longer fragments will have its fragment at the upper, will have its stain at the upper of the gel, and the smaller fragments, as their size is smaller, they will move downwards more quickly, and their fragment will be shown at the lower side of the electrophoretic gel. Now the sequence is read the sequence of the newly synthesized strand complementary to the original DNA template, and these stains on the fragment on the gel, it shows the length of the fragment. So we know that in the first lane we load the sample which contain the dideoxy G. So first of all, we know that our primer, it contain the TAG. It contain the TAG. So the TAG is the first, it is inserted. The first base which is inserted, and it is at the lowest end. So the first base is the G. Now the second would be the T. After G there is the T. Then after T the fragment contain the C, and the after C our template will contain the G. After G there will be the A inserted, and at the end T is inserted. So by using the size of the fragment on the polyacrylamide gel electrophoresis, we manually calculate, we manually sequence that which type of the nucleotide must be present after that nucleotide. This is the technique, or these are the principles which was used by the human genome project. But as the human genome, it contain a very complex amount of the DNA, the chromosomes, they will have a large amount of the DNA. We use third generation, next generation and high performance and automatic computers in order to sequence the genes. This is just I use the manual sequencing, but originally there is the usage of high throughput technology which allow the rapid sequencing of the entire genome. So this was all about my today's lecture. Best wishes and good luck.